Okay, so let's talk about cellular respiration. Cellular respiration. This is how and why we use oxygen in our bodies, why we need to breathe it in. Plants also do it, but they take their oxygen that they make once they break down water in photosynthesis. We're going to look at a mitochondria. The mitochondria, like a chloroplast, has two membranes, which is going to be an interesting discussion for later. So there's a membrane, and then there's another membrane that has all kinds of folds in it. Often the mitochondria is drawn like this with folds. And so there's this inner membrane space here. And that inner membrane space is different, right? It's a space that's separated by a second membrane from an internal space that we call the matrix. No, it's not some fanciful Keanu Reeves movie. It's a secondary space inside the mitochondria. This is the matrix. So this matrix is important because, again, as always, when you're compartmentalizing, when you're separating, you're able to do two different things, have two different sets of conditions. And by having two different sets of conditions, you're able to control what's going on. You can have one side have a low pH, for instance, another side have a high pH. So again, this is the matrix. And this is an intermembrane space. All right. So that anatomy of the mitochondria being clear, let's take a look at what's happening with glucose when it comes into the cell. These are two really nice drawings of the process. Well, glucose gets in the cell. As you know, glucose is a large polar molecule, so it's not going to get through the membrane easily. It shows it in this diagram. I have a problem with what they've drawn because they have it drawn as if glucose were just going to come right through. Well, there's going to have a channel here, a glucose channel, that's going to allow glucose through the membrane. It's the only way glucose gets in the membrane. So to be fair, you have to have this channel that allows the sugar through the membrane. Otherwise, it doesn't happen. That's how insulin works. Insulin releases these channels into the membrane and allows for glucose to leave the blood. Remember that the blood is out here, or the interstitial fluid, or what outside the cell is out there, and the cytoplasm's in here, right? Or inside the cell, the cyto meaning cell. So now we got the glucose here. The very first thing we do is we have to trap the glucose in the cell. We don't want the cell to get glucose to get back out. So we put a phosphate on it so it can't fit through the channel anymore. It doesn't leave. Now it's stuck inside the cell. And we're going to do this process called glycolysis. And you'll see it's labeled here, glycolysis, okay? And the whole point of glycolysis is, and let's go through the word again, glyco, meaning sugar, lysis, to break apart. Now, glycolysis has many steps. There are a lot of different changes you have to do to take this 6-carbon sugar, a sugar molecule that, again, looks something like this when it's in a ring form. Right? Where this is your 1' and this is your 6'. You have an oxygen there. So that's your sugar. So we have to take this molecule and we have to break it down very neatly into a 3-carbon molecule, and each step changes it slightly. We add a phosphate. We actually have to put an ATP in. The first place we're going to use ATP is we're going to use ATP, and I want to remind you that ATP is adenine. It's A with 3 phosphates attached to it. And these phosphates themselves are not the energy, but the bond that they create by breaking the bond, adding this phosphate, taking this phosphate off and adding it to sugar, again, trying to trap sugar in glucose inside the cell. By adding this phosphate, you are giving the process energy as well. So now ATP turns into ADP. I guess I should keep this 3-phosphate. And sorry, my sister keeps texting me. Mute this. All right. So what we're doing here is every time we add we're going to add phosphates using these enzymes called kinases. In this case, it's a hexokinase, but kinase just means you're adding. When you see the word kinase ever, it just means adding a phosphate. For you guys.
It's for our video.
So kinase is going to add that phosphate, and there are all kinds of different kinases. Again, this is a hexokinase, but it's irrelevant. The point is that every different kind of kinase is going to have a fancy a name that comes before it that gives you a clue as to what it does. Kinase means it adds phosphate, so it's an enzyme that adds phosphate. And there's a bunch of enzymes in each of the steps. There's an isomerase that changes the shape of one thing to another. There's all these little steps, A and B and C, that are happening. And we're not going to get into those details. Could you close the door, please? Thank you. But eventually, you take... I gave you a pen. Oh, that one over there is locked? It wasn't a minute ago. I don't have my badge. I forgot it. Huh? I wish I knew where there were bags. Can you see, is Miss Bannicourt here? Could you ask her for her badge? Tell her I forgot mine. So the 6-carbon molecule split up into 2 3-carbon molecules, and one is called, it's irrelevant what it's called as far as you're concerned, but it's DHAP. Again, you don't have to know this, but the one molecule I did ask you to know was this glyceraldehyde. Phosphate. Very useful molecule. It's used a lot in a lot of different biochemical pathways in your body. It's used everywhere. In lipid synthesis and breaking down sugar. But in any case, what ends up happening is you're gonna keep changing the molecule, glyceraldehyde 3-phosphate, until you finally make this molecule, which is an important molecule and a key step to all of cellular respiration is this molecule called pyruvate. Now, when you're talking about the DAP and the glyceraldehyde, the DHAP is just a temporary stop for that other half of the 6 carbons, as DAP will quickly be changed to glyceraldehyde 3-phosphate so that it can join this pathway to finally get to pyruvate. Now we added ATP here in order to keep the sugar in here. ATP is also, or phosphate there. We also add a phosphate here at this step. Let me go ahead and keep the colors the same. Where ATP got turned into ADP here as well. So far we used 2 ATPs. Remember, ATP is our currency, and so we need to make sure that we're making ATP. And so far we've used 2 ATPs. That's not good news. We've used 2 ATPs actually, if you think about it. So you might as well put down -2. We've lost 2 ATPs up to this point. As we keep moving down, what is interesting is that at some point, again, I'm not getting into the details, but at some point you're going to take and use and get, or rather put in 2 NAD pluses. Remember from our activities and from earlier in the week and our discussions with photosynthesis, these kinds of molecules are electron carriers. And they're very important because they're the way we're really going to get some ATP out of this process. You can think of them as like a nitro booster. And what we'll produce is, with the NAD plus, is 2 NADHs and 2 NADHs. That's good enough. So what'll happen then, as you get these 2 NADHs, these NADHs are going to go right to the electron transport chain, and we'll look at that next. As you go a little further, you're going to get 2 ATP back. So over here, 2 ADPs get changed into 2 ATPs. Now, why is all this doubled? Because remember, we had 6 carbons. We changed them to 2 3-carbon molecules. This goes through and creates a 3-carbon. The next goes through and creates a 3-carbon. So for every 1 molecule of glucose, you're using 2 ATPs. You're in the investment phase. We call this the investment phase, right? Where you're investing 2 ATPs. And here you get some dividends, right? You're getting something back. You're getting 2 ATPs back. This goes off in the electron transport chain. And then you end up with 2 ATPs back again. So that's fantastic. Well, right before it turns into pyruvate, you get even 2 more ATPs. So now you have a profit. So you actually gained 4 ATPs, but you lost 2, so you have a net ATP of 2. So you gain 2 ATPs at this point. This is all in glycolysis. This is all happening in the cytoplasm. It's not in the mitochondria yet. This is called glycolysis, the breaking down of glucose. And here we've come to the decision point. We have pyruvate. Now the NADHs are going off to the mitochondria. That's not a problem. ATPs can be used in the cytoplasm or not. Wherever ATP is used, it is used. But that's just producing just 2 ATPs out of sugar. That's not enough to keep you alive. But there's a decision point. And I call this a decision point because now the decision is made not by mentally, not consciously, but depending on what resources are available. The decision is as follows. If there's oxygen present when you're breathing, when everything's fine, and you have oxygen gas present, then it goes to the electron transport chain, or into the mitochondria, to mitochondria, to do the next step of the process. If there's no oxygen present, right? So there's no oxygen present at this point, then it does something called fermentation. Your cells do fermentation. Fermentation is an emergency situation. We need some ATP, but we're not breathing. When does this happen? When you're going up the stairs in the morning, when you're running 2 miles, you're using a lot of ATP, using a lot of oxygen. You're you're breaking down a lot of sugar, but if you don't have enough oxygen to take care of all that sugar, then you're running out of oxygen. If there's not enough oxygen present, so it doesn't have to be that someone's choking you to death, which is also, or that you're underneath the ice, you fell through a lake, you're drowning. That's not necessarily the only reason that you would do fermentation. It's also when you're doing aerobic exercise. You're pushing yourself to the point where you don't have enough oxygen for the need of your body, so you produce this thing called fermentation. Fermentation is going to take that. Fermentation, again, it's a stopgap measure. It's a way of trying to get some ATP out of a bad situation without oxygen. So without oxygen, this whole thing stops, right? It's like a conveyor belt. When you shut down the conveyor belt, if you're making you're breaking down sugar every second. If you don't have enough NAD pluses, or if you don't have enough ADPs to make ATPs, then you can't make ATPs. And this process doesn't work, so everything stops. You get backed up like a traffic jam. So the bottom line is that you need to have ADP and NAD plus available so that this can continue to go on, so that you can make ATP. In order to do that, you need to do a little fermentation. If you don't have oxygen, in order to do that, you need to do something called fermentation. So what you'll do is you'll take those 2 pyruvates that are stuck, right? Because you don't have any oxygen, so they can't go down to the mitochondria. So what you're going to do is you're going to take those 2 pyruvates and they're going to go, and you're going to take 2 NADHs that you made, the 2 NADHs you made up here, right? The 2 NADHs you made up here. You're going to use them here, and you're going to make 2 NAD pluses. Now, because now you have the 2 NAD pluses, now you can make the NADH, and now this can continue. You continue to make 4 ATPs at a time. So you continue to make some ATP, and by the way, you produce something called 2-lactate. And lactate turns into lactic acid, and you can't have too much lactic acid in your body. Not only is it bad that you're not making enough ATP, you're just making enough just barely to stay alive, you're also producing lactic acid, which is not very good for you. But when you finally start breathing again, once you reach the top of the stairs, you stop running or you come out from your underwater swim, you take that deep breath. Now you have oxygen. Now this all corrects itself. The lactic acid gets taken care of. The pyruvates can now go to the mitochondria and produce ATP. Then you can breathe out CO2. But until then, this whole process stops. Now we're going to go ahead and talk about what happens to the mitochondria next, but I'm going to stop this video here so that you guys, because I know you guys don't like to see 1-hour videos, even though I break them up into chapters. So there you have it. This is glycolysis.